It's my great pleasure to introduce Dr. Valentin Bruttel. He studied biomedicine and molecular medicine in the beautiful city of Würzburg and earned his doctorate in tumor immunology. Today he's Chief Scientific Officer at Toleris Biotherapeutics, working as a bioengineer and as an immunologist. On the private side, he's been very engaged in scientifically investigating the origin of the SARS-CoV-2 virus. And he's also on the advisory board of BiosafetyNow, a non-profit organization advocating for more lab safety. And this organization also supported the congressional inquiry in the United States. And since this promises to be a very exciting, very suspenseful talk, I don't want to keep you waiting any longer and hand over to Valentin. Stage is yours.
Awesome. You can hear me? Yeah. Perfect. All right. Thanks a lot for the great introduction. I can skip almost my entire introduction slide on my background. So what we do as my day job is develop biotherapeutics for autoimmune diseases. We discovered a mechanism in pregnancy where the embryo teaches the maternal immune system to tolerate it selectively without shutting down the entire immune system. We want to adapt that to autoimmune diseases and actually also to long COVID and ME/CFS. I'm the board member of BiosafetyNow, so there's some interest. I'm very interested in keeping labs safe and preventing pandemics, but I have no financial conflicts of interest, no virology projects or collaborations or anything like that. Very important, this is my private opinion, my personal assessment, not representing any of my employers, and this was investigated basically in my free time over the last 3 years. So why does the origin of SARS-CoV-2 matter? If we compare it to 2 world wars, I hope this works, yeah. Actually, we can see that it sits with regards to the cost that this pandemic caused right in the middle between World War I and II, and it also caused more deaths than World War I, according to a recent World Health Organization estimate. I don't want to go into details of how much of that was directly virus associated or caused by other problems caused by the pandemic, but it is of tremendous importance that we prevent something like that from happening again, and for that I think we need to know how it happened. This was also already taken out of me. Of course, new technologies bring new risks, which could be difficult to control. Why I choose this wizard's apprentice theme here. And what we are currently told about the origin is very, very diverse. So if you look, for example, at the European Union page, any manipulation of this virus or artificial creation is still put in the context of conspiracy theories. They say that no one is responsible. If we look at, for example, the White House.gov page, then this is called a lab leak. If we ask virologists very early on, they said that they strongly condemn conspiracy theories suggesting that this has a non-natural origin. And now very recent we have the United States Attorney's Office starting the first case against an employee of a funding agency who is actually charged with conspiracy against the United States. So it's a lot of framing in both directions. And unfortunately, in my view, very, very little evidence. So this is what I want to focus on today, just what I think is the important evidence without that many judgments. I think we have enough of that. Sorry, I need to use this. So let's just put ourselves in this situation. Imagine we have an airborne pathogen that kills many near a large biosafety lab. We have the Communist Party that blames contaminated meat. We have livestock that is killed and Western experts that don't get access to the labs actually, but they still confirm a natural origin. And everyone assumes that this is correct until something happens. And in this case, it was 13 years later, Boris Yeltsin admitted that this was a lab origin, which is actually the Sverdlovsk anthrax leak in 1979, which in my view has some similarities with the story in the COVID origin. And what we can learn from that is that governments are not necessarily always completely honest. We've also seen that with the Iraq weapons of mass destruction claims, which were not true. Some experts can be sloppy or conflicted, right? The literature cannot always be trusted, but the evidence actually was very, very clear. Yeah. Back then, all the cases clustered downwind of that facility where they had changed the filter and then had to stop the work and put up a note, which wasn't seen by the team next day. So they accidentally released these spores of anthrax. Then when you look at the patients, the lungs were affected, not the intestines, which speaks for an inhaled virus or an inhaled pathogen, not something that you eat. And if you look at the lab strain, the strain that caused the pandemic was highly related to a typical lab strain. So the evidence can be very clear, but the conclusions can be quite divergent. And so the question is, can we trust the experts? I think it's a bit difficult when there are conflicts of interest at play. For example, in the child abuse scandal in the Catholic Church, there are probably thousands of priests or bishops that knew that something was going on. Not one of them really spoke up and alarmed the public. VW cheat devices, a little bit similar. First evidence of cheating was from the 1990s, and almost to the last day, they kind of denied that and said this was like technical glitches in the measurements. Yeah. So I think we should trust the facts, which is what I try to do here. But to assess those and to assess the conflicts of interest, we need to go a little bit back and look at how synthetic viruses are made. Actually, this technology was pioneered in the year 2000 by Ralph Baric in the United States, who produced the first SARS similar genetic virus. And this is done by putting this huge genome of 30,000 letters, 30,000 nucleotides, into smaller segments that can be easily handled and produced. And here they use these junctions. And to produce these junctions, they use restriction enzymes that act like scissors. They have recognition sites at which they specifically cut. So in this case, the GCC and then GGC sequence. And they use some of those that produce different sticky ends. So these sticky ends in the DNA are single-strand overhangs that kind of stick together. And then you can glue them together with an enzyme called ligase. And because they are all different at the different junctions, you can have the entire virus reassembled in the correct order. So this is basically the breakthrough technology. And these restriction enzymes, most of them produce always the same sticky ends, which is not very good. You end up with segments that could form circles or arranged in the same, in the wrong order. But if you use enzymes like these, you get always different sticky ends and you get the correct sequence. Now, how do you introduce these restriction sites is by silent mutations. So there are two different types of mutations in biology. Some affect only the DNA or RNA code, and some also change the protein sequence. And you don't want to change the protein sequence, otherwise all your colleagues will say, oh, this virus is not functional because you put in a new protein. But with these silent mutations, so taking a different DNA, RNA sequence that codes for the same enzyme, you can introduce or remove these restriction sites without changing the function of the virus. And then what they did is they amplified these individual segments first, then digested them via these gene scissors, and then assembled everything into one big, large DNA construct called a bacterial artificial chromosome. And this can then be put into a cell that is highly susceptible, that doesn't have important defense mechanisms against this. And if you put this DNA segment into the cell does everything else. It transcribes to RNA, the RNA is transcribed into proteins or translated to proteins, and then the proteins are assembled with the DNA, or the RNA in this case, and the membrane of the cell into an infectious virus. So it's a very simple technology. Just from code, you get something that can be lethal. And the technology is now so advanced that such a virus can be built for like material cost of $5,000 within 1 week. So if you compare that to nuclear weapons, which are also very risky and highly regulated, it's probably 1,000 times more expensive, or at least, I would say, I don't know. And it requires significantly more money. And this is why I think this technology should be strictly regulated. So what happens in Then later is that actually other virologists are still breeding viruses. These are not completely assembled, they're just adapted to humans. And these were referred to as nightmare or killer viruses. These were bird flu viruses that are 100 times more lethal than SARS-CoV-2. And they adapted them in a way so that they could spread via the air from human to humans. So highly risky experiments, which led to what I often refer to as the war within virology about what can be done, what should be done, which is probably a debate you're familiar with in the context of AI. So on the one hand, we had the Cambridge Working Group. They asked that we should pause the creation of these potential pandemic pathogens until we can completely assess the risk. And then on the other side, we had a group called Scientists for Science. These two gentlemen, Fouchier and Kawaoka, were the ones who built these nightmare viruses. And then Christian Drosten was also involved here early on. And they said that we should focus on lab safety. This is enough. And we should not limit what they say. They were opposed to limiting the types of experiments. And then there was, in my view, lobbyism. So, for example, in Germany, some virologists asked that we have to introduce risks. In the Frankfurter Allgemeine, in the United States, we had Anthony Fauci and Francis Collins, the heads of the NIAID and the NIH, so the equivalent of the DFG, with a combined budget of more than $40 billion per year that they put into research. So almost every scientist is dependent on these people in the United States. That said that this risk is worth taking. So these experiments are needed. So there still was a pause on funding for these risky experiments put in place under Obama. But this led to what I think is a movement to China, where this was not really regulated. So Ralph Baric started collaborating with Xixing Li from the Wuhan Institute of Virology. They made the first viruses in China. This is the segment map down here. And then these stars actually indicate the individual mutations, silent mutations that are introduced to add or remove these restriction sites that you need for the assembly and to generate these DNA segments. Only a year later, the first, say, purely Chinese viruses were made independently of US efforts. They used now better restriction enzymes that produce longer sticky ends, so they work more reliably. And then again, almost only 1 year later, they made the first experiments where they put in receptors from other viruses into harmless bat coronaviruses, which were predicted or assumed to be better adapted to infect human cells. And then they tested the infection capacity in mice that have the human receptor. So these are transgenic mice. And at some time points, they had a 10,000-fold higher viral load by putting in these new viruses. So the debate in the United States changed. They said, oh, gain of function is only the case when you mutate viruses that already have caused pandemics or that can already infect humans. And these bad viruses are not gain of function experiments. In my view, this is clearly gain of function. And ours, like the cautious side, kind of lost this debate in this case. The ban on the funding was lifted, and then also like Peter Daszak, who led this organization in New York, EcoHealth Alliance, that brought a lot of money to China, got his funding for gain of function research reinstated. In 2018, we had this diffuse draft that was already mentioned, an application for a grant submitted to DARPA, and this is very important. This was actually leaked later by a DARPA employee. All the scientists involved tried to cover this up. It involved, again, about scientists that we already know, Peter Daszak, Ralph Baric, and Xixing Li. And this was not funded, but very similar experiments were. And we now, since just last week, know that Baric also received funding from Europe, from the Wellcome Trust around that time. And he might get into legal trouble because he didn't declare that for whatever reason. Yeah. But we don't know what was directly done in these other grants because we don't have the complete documentation, unfortunately. So I mostly refer to the diffuse grant, which in my view is the key piece of lab origin evidence. And I find it very questionable that all the applicants, all the institutions, all the universities involved did not come out and say, look, we did this. This was our plan. We never did it. Look at our lab books. Nothing to hide. Yeah, but try to cover this up. Of course, the initial publication said that this is a natural virus. For example, we have this most cited publication called The Proximal Origin of SARS-CoV-2. Their key arguments were that this is a new receptor binding domain. We've never seen this before, and a new virus. So they would have more likely used an already established viral backbone, which of course can be the case, but of course also every new genetic backbone is sometime published for the first time. And if you cause a pandemic before, you're not going to publish the virus 2 years later. So it's, in my view, very weak arguments. And now we know what they actually debated in their emails, which is quite interesting because the head author told here, Anthony Fauci, that he and his two co-authors all find this virus inconsistent with expectations of natural evolution. So they said this doesn't look naturally evolved. We know another of the authors says this doesn't really exclude an inadvertent release, and that there is a nightmare of circumstantial evidence that has to be assessed. And we also know that actually the funders, so Anthony Fauci and Farrar from the Wellcome Trust, had a big impact on this publication. They wanted changes, and they supported and put pressure on the journals that published this. So the emails tell us actually a lot. But let's focus more on the science-heavy publications that claim a natural origin. First one was this one called the Molecular Epidemiology. This focused on two lineages, lineage A and B, that were both found at the market in Wuhan. And of course, one point where they're correct is that a lot of these early samples cluster around the market. Many were found at the market. Big problem is these lineages differ only by 2 mutations. We know from that case at the Diamond Princess cruise ship that up to 3 mutations can occur in a single patient. So this is not really something that came to humans with a lot of difference in time. And it's just impossible to say if a mutation evolves in an animal or in a human. These are not animal-specific mutations. So they could have jumped twice from a patient to different humans or twice from an animal to different humans. It's just impossible to know. So it's very questionable with regards to the conclusions. They also ignored some of the intermediate genomes. And we also know that lab leaks have actually leaked several lineages before. For example, in the Sverdlovsk anthrax leak, a huge amount of pathogen was released, and that infected many people. And whenever you have a huge amount, the risk that you have several different mutations is much higher. Yeah? So from in a human-to-human transmission, it's probably just one droplet with a few thousand viruses. Most likely, most of them are identical. If you leak a few liters of virus supernatant or something like that, of course, there can be many mutations in one lab leak. And then also very recently, there were severe coding errors in this paper, which actually now hints that this came from a single mutation, from a single origin. Then the next paper discussing the origin in science was more focusing on the proximity of the samples to stalls that sold, for example, animals at the Wuhan market, and the distance to the famous biosafety level 4 lab in Wuhan, which is across the river in the very south of the city. The major problems here is that all the samples that were tested that we know of that came from animals were negative for the virus, and that we already had thousands of different cases in humans around that time, according to Peter M. Barak from Denmark, who was in the World Health Organization Commission, and that there was the sampling bias. So they specifically looked for patients that had exposure to the Wuhan market and then sequenced them. So of course, this leads to finding more patients from the market. And then there was some issues with deleted data by the NIH, not very nice things. And another trick they did is they simply showed the animal stalls, but for example, didn't show the toilets, which is a very enclosed space, right? You had this very open hall where viral loads can completely be removed or washed, blown out by the wind. In a toilet, you have this very many people in a very enclosed space. You could have higher viral loads because of that. And there also was this gambling room that was poorly ventilated just on top of that. So it could have been all from humans. And they didn't show the biosafety level 2 labs, which is problematic because we know today that when they asked Xixing Li at which biosafety level they did the experiments, she said at level 2 and 3. So if you just refer to with the biosafety level 4 lab, which is far away, it doesn't really help. If you add the biosafety level 2 labs to the map, you get some that are right in the center of this outbreak. Yeah? Biosafety level 2 is comparable to a dentist's praxis, to a dentist's workplace. You have a lab coat, you have gloves, you have goggles, but you don't even have to wear these simple face masks that we had to wear when we had to shop at Aldi, for example, for a few months. Biosafety level 3 and 4 are a completely different story. You have this pressure suits. In biosafety level 4, the entire lab is enclosed in air-filtered enclosure. So doing these experiments of biosafety level 2 is very, very risky in my view. There were other severe safety concerns, for example, failures in safety equipment. I can't go through all the details, but there were 6 of them that are mentioned in this report just within half a year before the outbreak. And then actually it was a report done with 60 Minutes, a US TV show, where they visited the lab, invited by the virologists there. And if you zoom in, you can see that they kept their coffee mugs directly on their workbenches, which is an absolute no-go. There are bicycles in the lab in different... So it's really a nightmare. This was when they invited TV to the lab. I don't want to know how it looks like otherwise. So anyway, let's focus on the more robust origin evidence. So first of all, I think the outbreak location is important. Of course, this was very obvious from early on. So the most similar viruses called Reggie 13 and Banal 52 were found actually very far away from Wuhan, about 1,500 kilometers. In regions where you have these bats that harbor these viruses. Yeah. So the key question is, how did these viruses get to Wuhan without leaving any trace? Yeah. And if you assume that this could have been a lab origin, what I did here is just look for the number of citations of Chinese megacities combined with bat and coronavirus, and you can immediately see that Wuhan published way, way more about bat coronaviruses than any other related city, especially if you normalize it to the population. So a lab origin you would assume to happen in Wuhan, a natural origin you would assume to happen in Laos or maybe Yunnan. Yeah. Then again, if you look at the diffuse proposal, they described there in detail that they had more than 180 already sequenced coronaviruses in the lab and more than 10,000 bat samples. And that they had sampling sites in Yunnan and Laos, and that they shipped these viruses always to the Wuhan Institute of Virology. So another big issue was that they took their databases offline in the fall of 2019. They say this was due to hacking, that they had problems with hacking. I don't know why this was the case for 6 years on, but there were actually several different databases. This was in detail investigated by a group called Drastic, independent scientists from all over the world who did a lot of lab origin investigations. And this would just be the perfect explanation how these viruses got from Yunnan or Laos to Wuhan. If you had either an infected researcher that maybe didn't even notice the infection because it was very mild in many of us, right? Or you had a sealed sample put in a plastic box that is then just shipped. Yeah. The other big thing that jumped out immediately was how well this virus was adapted to human spread from the get-go. Yeah. So the comparison here is, for example, the green line of SARS-1, where you needed 8 months to get to 8,000 patients, and then MERS is here. You needed 12 months to get to 100 patients, compared to SARS-CoV-2, where you basically have almost a vertical line. And this is very unusual for a natural virus, which usually needs a lot of time to adapt to the human receptors. And during that time, you have very slow spread. But if you look at, for example, molecular analysis of the receptor binding domain of the spike protein, then this is predicted to bind perfectly to humans, much better than to, for example, cats or bats or dogs or pangolins, which is a bit suspicious. This should have been initially adapted for an intermediate species. And again, if you look at the diffuse proposal, what they say there is they want to basically model interactions between different bad coronavirus receptors and the human ACE2 gene. They also want to screen for spike proteins that can interact with an additional human receptor, which was also found to interact with SARS-CoV-2. And of course, if you model and screen spike proteins for perfect binding to humans, and then you leak something like that, it's no surprise that this spreads like wildfire. If you actually look at the precise figures from their proposal, they mention the sequence of the receptor binding domain they want to modify. And if you compare that part, the receptor binding domain, with the highly related Regi 13 virus, you see this red area. There are about 20% mutations in that area, while the rest of the genome is almost completely white, 1 to 3% mutations, right? So it looks like a replaced site in at least one of these viruses. The other side that sticks out is this fusion cleavage site, which I'll talk about now. So this was in the media for a little bit. A fusion cleavage site is something that enables the virus to infect different cells in the body. So the virus sticks with these spike proteins to the receptor, to ACE2, but this doesn't help yet. The virus also needs to put its genome into the cell, and for that, it actually needs to cleave off part of that spike protein, which can be done, for example, via trypsin, which is a protease found in the intestines. This leads to diarrhea and not so dangerous infections because it's very difficult to spread among humans. But if you put in furin, for example, which is also expressed in the lungs, you can have much better spreading via the air from human to human. And ACE2 is also expressed, for example, in the brain, which can lead to very severe, much more severe outcomes. And actually, there was this kind of, I'd also almost say, trend in gain-of-function virology since 2006 that they put these fusion cleavage sites into all kinds of viruses. For example, here in SARS-1, then there's one example where they put this in a porcine epidemic diarrhea virus, and then one where they put it into a MERS virus. This was done by Barrick and Xixing Li. And then also into these bird flu viruses, which were extremely lethal. Just to give you an overview, the lethality of most of these viruses is between 20 and almost 100% in piglets, while SARS-CoV-2 is much, much less dangerous, below 1% case fatality rate. So these are really dangerous experiments that were mostly done in Europe and the United States. And this is just one example. When you remove the fusion cleavage site from SARS-CoV-2, for example, in hamsters, they don't get sick anymore. So it seems to be really a key, let's say, key to enable viruses to spread into other hosts and to infect highly vital organs. If you look at the fusion cleavage sites, where it sits in the SARS-CoV-2 virus, and you look at related viruses, all these are the closest relatives, you can immediately see that SARS-CoV-2 is really unique with having that fusion cleavage site, and looks like an inserted fusion cleavage site, which can happen naturally. But again, if you ask what is proposed in diffuse, they have this graph where they suggest to put in fewer cleavage sites into the S1, S2 border, which is exactly this location. So this R is already there. You need 2 Rs for the fusion cleavage site. So this is basically the only place you can put it in if you follow this proposal. Yeah. In a 30,000 DNA letter or RNA letter genome, it's exactly at the site that they propose to put them in, if they don't find them. And then there's one more point. They said they want to use human identical fusion cleavage sites, which is a bit weird because they're very similar. Actually, fusion is in many hosts, and but if you look at the complete sequence of the fusion cleavage sites, it completely matches this human enzyme that was studied at Ralph Barrick's university. So it could be the template. Yeah. Then there's the issue of no intermediate hosts. We still haven't found any animals that are known to harbor this virus or have an ancestral virus. These were found very quickly in the examples of SARS and MERS. There were some articles that claimed raccoon dogs were likely intermediate hosts, but if you look at the DNA in detail, you see actually a negative correlation here. So the more raccoon dog DNA you have, the less SARS-CoV-2 RNA you have, which speaks against this host opposite case when you look at human. And then there was one paper that tested this. They put lots of RNA of these, lots of viruses into the nose of the raccoon dogs, and they got a short infection, but the viral load was about 10,000-fold lower than in humans, which is just speaking against these being good intermediate hosts. And actually, there was an ancestor found in a cell line, the so-called Vero cell line that I mentioned already, in accidentally leaked contaminating reads that were released with another experiment. And some of these viruses here had exactly the sequence you would expect from ancestral virus, because they have this TTC, which is found in the natural viruses and predicted for the ancestor, and which is no longer found in these two lineages that were found at the market. So the virus samples could very likely be actually a sample from the most ancestral human genome. And actually, when you again look at the diffuse proposal, they mentioned that they want to use these Vero cells and that they want to sequence their DNA at exactly the company that sequenced this DNA. We also know that I probably don't have time to go into this in detail, they already uploaded a part of a 99.5% protein identical virus in 2018 to NIH servers, which is why they also had to then later release the rest of the RITG13 genome, which is much more divergent. But this puts them basically, most of these most related viruses were actually handled by the Wuhan Institute of Virology. And then there was an interesting experiment with artificial intelligence. A guy in Spain programmed AI to search all the huge DNA databases for SARS-CoV-2-like sequences, and he found a 100% spike identical match, but with a process called codon optimization. So the RNA sequence is very different, optimized for expression. And these samples were taken in 2019, when officially no one knew what the sequence of this virus was like. And they were actually also containing a trimerization domain. So this is a domain you add to generate a vaccine that can work independent of a membrane. In the membrane, these spike proteins automatically trimerize. If you want to use this as a powder vaccine, of course, so you need these trimerization domains to have them in the correct conformation. Again, if you look at diffuse, they wanted to create codon-optimized spike trimers and use these gel or powder vaccines for bats. So this would exactly fit to their proposal. But to end on the evidence, this is actually my own work. We looked at these restriction enzymes, these gene scissors, together with Tony Van Dong and Alex Washburn. And to briefly recap, we looked at how these viruses were built before. It was mostly these three gene scissors that were used, and these BSA1, BSA, and B2 were used in 2017 by the Wuhan Institute of Virology. They often use these 5 to 8 segments, the longest segment usually below 8 kb, and then the unique sticky end. So if you look at the upper example here is the natural MERS virus. You have just 2 restriction sites, huge fragments. If you look at the lab variant, you have these fairly even spaced segments, and the same is the case with this Wuhan Institute of Virology virus 1. You have very unusual spacing, and then with the lab virus below, you have this very homogeneous spacing. So you have this Goldilocks zone, so to say, of 5 to 8 fragments, with the longest fragment being only about like 30% of the viral genome in length. And then if you look at SARS-CoV-2 here again, compared to 10 other lab-made viruses, it sits right in that window. This is not yet the strongest evidence. We assume about 1% of natural viruses also have that pattern. But SARS-CoV-2 is a bit different because it has these 2 unique BSA1 sites that allow for efficient replacement of the receptor binding domain at the fusion cleavage site, which is exactly the experiments they wanted to do. And the key evidence, and unfortunately you can read a lot about our publication in almost every German newspaper. Most of it is very negative, but you will never find anyone mentioning our key evidence. So the key evidence is shown by the lowest p-value, which indicates how strong the evidence is, how likely it is that this is of natural origin. And when we looked at the frequency of mutations in these stitching sites, what we assumed is to find a 3% mutation rate, but what we found is 22%. So the chance that this is natural, especially in comparison to the Reg13 virus, is far below 1 in a million. And I'm super happy to discuss this with anyone. No virologist so far was willing to discuss this. Yeah, if you see articles of our paper, maybe ask the journalist why he didn't mention our most important finding. And then, as we already heard before, 2 years after our preprint, which unfortunately was rejected by most journals, they didn't want to send it out for review. And then there was this draft of the diffuse proposal leaked, which mentioned, as we heard before, exactly these segments and ordering BSMB1. So now the odds are even lower than before. Yeah. So I just want to summarize really quickly. I have to come to an end. There are, in my view, circumstantial pieces of evidence and then molecular pieces of evidence. I wanted to ask how consistent they are with labs or people or experiments planned in the diffuse proposal. And I think that we went through most of them. There was this issue with lab safety. We know that the outbreak started near a safety level 2 lab where they did these experiments. Actually, it was also surprising that they shut down pretty much the Wuhan Institute of Virology was taken over by government officials, and virologists were no longer to communicate, which is a bit weird in an outbreak, or that should have been their expertise. There were suspicious emails, and dishonest authors went through that. The deleted databases, eyewitness statements, I didn't have time for that. And then the timing and the location. Yeah. So these viruses circle for thousands of years. The bat caves are very far away. Sick researchers, according to the US intelligence. And then if you look at the molecular evidence, I don't know how much we can trust intelligence, but molecular evidence I know more about. So we know that both these receptors they wanted to use work for SARS-CoV-2. We know that they had these related viruses synthetically assembled. The Reg13 sequence is very weird. They mentioned the furin cleavage site, which is unique, the first time in this family of 1,500 viruses. They mention the Vero cell line to produce these, which is then sequenced at the company, Sangon. And the plasmids are something you would use to do these kind of experiments, characterize receptor binding with just the protein and not the exact virus for what you need these plasmids that have been leaked into these samples. And then, of course, the restriction site pattern and the mutation frequency are also very suspicious, and I just don't know any natural explanation for that. So due to that, my conclusion is that a lab origin is now proven beyond reasonable doubt. I'm very, very open to discussing this and to opposing evidence, which has been difficult. But I think that, no, I don't have time to go over the personal consequences, but I'd really like to thank most of the people who investigated all this. Hardly any of that was done by me. I want to thank my family who went through a lot in this time, and then my co-authors. And with that, I have to come to an end. And I think that we should really stop enhancing human pathogens. Otherwise, human pathogens may end us. With that, I'd like to have some time for questions. And thank you very much for your time and attention.
Thank you very much for your talk. I guess there are some questions somewhere in this room here.
I was quite happy at the beginning to see that there was a fellow immunologist here. I have to say I'm quite sad after your talk. Because, I mean, you showed bits and pieces of basic molecular biology somewhere, but honestly, as a molecular biologist, as an immunologist, I'm not really convinced. And before discussing all the biology stuff, which maybe is not the place, what would you like people to do now? So you showed a BSL-2 lab. You've probably worked daily. I work daily in a BSL-2 lab. The pictures that you showed are not the places where usually infection experiments happen, for example, because we do that in flow hoods, right? That's where we usually do the experiments. But apart from that, what would you propose? Because a lot of these viruses, for example, can infect cells, but they are modified in a way when we do the experiments that they cannot like multiply themselves, right, and go to the next generation. That's usually how we do experiments in the lab. So in your view, what do we need to do now? What do we need to adjust so that the experiments can be safe, or where do you think the errors are?
So in my view, we should go back to banning making human pathogens more lethal or more infectious for the time being, until we can really, really be sure that this is done under very safe conditions. Often the knowledge gained is also not so great. You can do this with, for example, artificial viruses that cannot infect humans anymore. If you have specific critiques on the evidence, all the references, all the data sources are on my Substack. You can just put in my name and find that. I'm very happy also to meet afterwards, or I'm very open to any criticism if you find anything wrong with the data. And I think we just have to at least be responsible enough to not repeat this accident, which means we should have an international body like the International Agency for Nuclear Energy, for example, that supervises nuclear weapons, that regulates this all over the world, that goes into the labs. We could have a lot of other instruments like registering all gain-of-function experiments. We could put in codes into the virus that tell us immediately, okay, this is a lab virus. There could be a lot of things done, but to gain the political momentum to do that, I think we first have to be honest about what caused this pandemic. Yeah? I think it's very difficult. People don't want to talk about it. It was not a pleasant time. And I don't think we can pressure the big countries that funded this or did these experiments to concede and come together, because this would be kind of an admission that something went wrong, right? So my way is to just speak the truth, what I think is the truth, and to warn fellow humans that this is dangerous stuff. They had very little resources, like $5,000 in a week in a lab is enough to cause something like this. And this is why I should, in my view, we should regulate this. But of course, if people think differently, I can't. I have to accept that.
Yeah, there's one question from Zoom. What was the goal of the diffuse proposal?
Yeah, so their key argument always was to be able to predict how close natural viruses are to causing pandemics. Or in this case, actually, this was around involving the military. So they said, oh, if you have to do some military operations Southeast Asia, there's the risk that our Marines get infected. So we want to evolve these vaccines, for example, to lower the viral loads in bats, so we don't get the military infected, for example. And the general theme in gain of function is always, we need to know how dangerous is the virus. So we do it in lab. We introduce 1, 2, 3, 4 mutations, and then we see is the virus then able to infect these humanized mice or model animals like ferrets that are very similar to humans. And then we know how dangerous this is, and maybe we can take countermeasurements. Yeah. So this is always the argument. There's also lots of money flowing into this, and you can publish this very easily. Some of these experiments in the horror viruses were just done by infecting 5 ferrets, always taking the virus from one ferret and putting it into another. And this then results in a science publication, which of course can give you a tenure for life. So it's very attractive for the virologist. It's not technically that challenging. Of course, you need this high safety labs, which are very expensive. But that, in my view, also brings in a bad incentive to go for more risky things, because the advantage for the individual scientist is clearly there.
Well, first of all, thank you for speaking about this topic openly. We all know what it could mean in this media environment. But now that the virus is circulating in the population, there has been a lot of work in the recent years towards the effects or long-term detrimental effects on the population. What do you think about that? And maybe specifically because you have a background in tumor biology, what do you think about the possible carcinogenic effects of SARS-CoV-2?
Mm-hmm. Yeah, so the virus is still circulating. You still have infections. You still have the risk of developing also severe disease like long COVID and ME/CFS, which doesn't disappear and which is really, really detrimental, I think more important than cancer in this context. This is there with every further infection that you get. You can still develop ME/CFS, which binds these poor patients to the bed and basically incapacitates them, or like enables them to do nothing except for like having a talk for like half an hour a day or something. It's really detrimental. Related to cancer, I don't see a very clear signal, but I haven't studied this in complete detail. I'm now working more in autoimmunity. Yeah, so there are more cancer cases for some tumors, but I don't know if this is directly associated with the virus. Some say it's associated with the vaccine, for which the evidence is also not so clear in my view, but there could also be potential risks. Yeah, but I can't have a conclusive answer on that, unfortunately.
First of all, thanks for the talk. It was really interesting. I was wondering, since you stand so hardly against such experiments, but at the same time you mentioned that experiments like this can be replicated with $5,000 and a week of time. How would a ban on such things help? Because time has shown that prohibition does not work in multiple areas. How would such a ban affect or help with such a case?
Yeah, of course it's difficult. What you could do is, for example, look at the companies that produce these DNA segments and make it illegal to produce segments of human pathogens, for example, which would make it much, much more expensive, because most labs actually, because this has become so cheap, they lost the capacity to make DNA themselves, which was done maybe like 20, 30 years ago in many, many labs, but it's not done anymore. And then, in my view, it had worked with nuclear weapons, right? Apart from the 1945 explosions, we didn't have any nukes going off. So, of course, it's difficult. It's way more difficult to control it than nukes. But if you have transparency, and you also need money always. This is not really industry focused, in my view. These experiments are mostly funded by governments. So you have these centralized organizations that could just say, okay, we don't fund these experiments anymore, which would be already a major step, I think. Which I think also should be done in Germany, actually. There's no ban in the US and in England on gain of function, dangerous gain of function. There's also very harmless gain of function experiments, which is not human pathogens. You just put in a gene that makes a bacteria glow green, so you can see what it does if it dies. This is not problematic at all, of course, but dangerous gain of function with human pathogens and that increases lethality, I think, should be made illegal and made illegal to fund with our tax money.
I'm not a biologist, so I have no comment on the second part, but I was wondering where you have the emails from and who claims that these email contacts are legit.
Actually, there's a lot of journalists in the United States who use this tool called Freedom of Information Act, so FOIA lawsuits. And as all of these scientists are employed by the government, there's this law in the United States. In theory, it also exists in Germany, but it doesn't really function in my experience. But in the US, you can just sue the university, say this is of public interest. We think one of the government employees was involved in something that maybe endangers the public, and then they have to release the work emails of these virologists, at least to some extent. So again, if you look at my Substack, this is all linked in there. And it was really, to me personally, because I'm a scientist, I believe in science, was very, very sad and shocking to see that these professors from the West and from independent institutions all almost believe the opposite to be true of what they published. Yeah.
Same for me. I'm not a biologist, but thank you for your speech. I'm really interesting. I mean, just believing what you showed and believing in science. Why would the public, free journalism, not be interested in digging more into that? Why? I mean, I understand that governments might not be interested, but I think there's still some free journalism around the world. Why are they not picking that up?
Yeah, that's a good question. Why it's not happening in Germany? Because I think we didn't fund any of that, and we should be the freest. I think so. there is very good journalism in the United States. There are really, for example, US Right to Know, an organization that did a lot of these FOIA lawsuits. They published everything and they really went deeply into this. And there are politicians there that then investigate virologists in these government hearings that know all the enzymes and that know all the technology. And there's a lot of involvement also from politicians in the United States. Here, unfortunately not. I think we have also, to some extent, a conflict of interest with science researchers because they're highly dependent on scientists being willing to interact with them regularly, right? So if you accuse someone of having caused something terrible like that, question is, will he or his colleague give you an interview 2 weeks later when you have to publish the next segment about whatever the latest Nature publication? And it is just very difficult to write all these science journal pieces completely by yourself because you have to go very deep into the science, which takes a lot of time. So it's always the safe option to just call the scientist and ask for his opinion and then quote him, and then you can put it out there in a day. You write a nice piece. So I talked to one journalist from the Netherlands. I gave him a 2-hour interview, and then he wrote an article about this. He didn't mention any of that. Then I asked him why he skipped all the molecular evidence I told him. He said, like, yeah, I have to reach out to the virologists I'm dependent on them. I also wanted to bring them into the discussion, things like that. There's sometimes this overlap, yeah. I don't know. There's some free journalists in Germany also that report on this. Some newspapers, like the Berliner Zeitung, is more open on NZZ in the Züricher Zeitung. They report more about this. But yeah.
Commission, I think from the parliament, I'm not sure if it was the Bundestag, that are trying to find out the causes and etc. Is that closed? Is it not taking into account these things? Are they hiding up? I mean, is that not a forum where you could place things like that, right? And force or get politicians from, I don't know, from which party able to support that?
Yeah, so there was actually one party demanding that, which I'm not a big fan of, but most other traditional parties in Germany refused to have this Untersuchungsausschuss, so a true investigation where you can ask people to come and they put under oath and you can search computers and sources and so on. So what they did right now, they just re-investigated the response basically to the pandemic. The origin question is completely taken out. It's not allowed to talk about that. I think it's not going into a good way. And I don't know why so many politicians are so scared of that debate. I mean, for example, Australia immediately said we need an investigation into the lab origin after the outbreak, and then China shut them off. They didn't buy their ore, they didn't buy their steel. And then there was one investigative journalist, Aya Velasquez, who's now she just reported about this, but one guy sued to get these expert commission protocols open. And in that, we don't know who, but someone said that China is a competitor, but also a partner, economic partner. It's very important. I don't know what other things went on. We are highly dependent on China for like medicine, technology, everything. So I can only speculate. This is the only piece of evidence I know.
The question is slightly off topic, but we had a former Big Techday speaker, Ian Goodfellow, which is an absolute hero for the machine learning community. And, you know, I always try to look at what speakers do and whether they want to come again. And he has a severe case of long COVID. And it's like, literally is the hero. He was at Apple, at Google, and wrote the seminal book. And he can walk again now, leave his apartment apparently, but it took years and years. And so this is not so much an origin question, it's more an outlook question. Like, as the molecular biology of this genetic system has been understood so well, what are the chances of having actual fixes for the people who are still suffering?
Yeah, this is also regarding my main job, where we use this mechanism from pregnancy to induce immunological tolerance, because in long COVID and ME/CFS, it's known that people have autoantibodies, so immune response against so-called G protein-coupled receptors. So these are receptors that, for example, regulate the blood vessels if they contract, or they regulate the transmission of neural signals. So I think our technology could help there. We are trying to get grants, which hasn't been easy since all the negative press about me. We are trying, started the company, trying to complete a financing round. But apart from that, I've just been at the International MECFS conference in Berlin. There's some repurposing trials. Some bring some benefit, but some also with side effects. It's not super encouraging. Yeah. And some people recover themselves after a few years, so there can be hope, but especially for the very severe case, it would be great to be able to do more, yeah, and to prevent further outbreaks. Yeah.
Okay, with that, let's thank Valentin once again, also for the discussion.